Cancer is a multifactorial trait involving many protein-coding genes. For example, there are tumor suppressor genes like P53, often referred to as the guardian of the genome, and BRCA1, often referred to as the breast cancer gene, which protect us from cancer because they prevent cells from dividing when they shouldn't, say when their DNA has been damaged. So these genes prevent cancer. There are other genes which also protect us from cancer because they can engage a process known as apoptosis, or programmed cell death. If a cell begins to act inappropriately, it can be triggered to destroy itself through this programmed cell death. And there are multiple genes involved in this pathway. Of course, DNA does not only encode protein encoding genes. We know that while only 3% of the genome codes for the amino acid order of proteins, about half of the genome is transcribed, forming a transcriptome with many RNAs which are never converted into protein. These include microRNAs, which are important in the regulation of the mRNAs for genes, given that they can bind to mRNAs and then either cause their degradation or block their translation into protein. In this example, a microRNA is capable of recognizing the mRNA of P53, and after it binds, this P53 transcript can be destroyed. And so thus the cell would lose proteins which would potentially prevent it from advancing in the cell cycle. If such a microRNA was overexpressed, this could promote cancerous growth. In the same way, there are microRNAs which bind to the mRNAs for genes which encode the apoptotic proteins. So if these miRNAs are overexpressed, the cells make fewer proteins which could promote the programmed cell death of abnormal cells. Cancer cells can only succeed and proliferate if they can prevent apoptosis, if they can prevent the programmed cell death, which would eliminate these cells once they become abnormal. And so overexpressed microRNAs are known to have a role in multiple cancers.